Hello, my name is Emir Selimovic. I'm a PhD candidate for the Marine Beyond project. I'm based in Ireland, working with the Irish Agricultural Authority, CHAGAS, and the University College of Dublin. My work is focused on the multi-stressors effect of nitrates and pesticides on the ecology of a river. Specifically, I'm working with macroinvertebrates, widely used to measure ecological quality. My main objective is to establish ecologically relevant thresholds for nitrate and a widely used pesticide for chloro to methylphenoxyacetic acid, otherwise known as FCPA. These are among the most pressing pollutants in agricultural catchments, and there are still knowledge gaps on their combined effects on aquatic communities. Currently, thresholds mostly consider mortality as their endpoint metric. However, stress can induce shifts in macroinvertebrates community, producing cascading negative effects that impact the entirety of a river ecosystem. One of the objectives behind is to look at the effects and interactions of multiple pollutants in agricultural catchments, and my work fits into this framework, focusing on nitrates and MCPA. Current legislation overlooks combined effects, and this can cause unexpected implications on river health. To address this issue, I will be looking at what level of stress key aquatic bioindicators can withstand before affecting the other organisms which depend on them, disrupting the entire community. The aim is to produce data to better understand ecosystem dynamics and ensure good biodiversity and ecosystem resilience. To investigate my research questions, I'll use a series of microcosm and mesocosm experiments. These are controlled and reproducible ways to investigate the sublethal effects of pollutants. For the microcosm experiments, I will collect organisms with a high sensitivity to pollution, such as mayflies, in the field, and bring them back to the lab, where I can control their environment and test them. The innovative methodology that we'll be using involves measuring the level of oxidative stress biomarkers within the organisms. The results will be used to inform the next experiment, which will be to implement a mesocosm. This phase will bring the study to a higher level, looking at the dynamics and composition of the entire community, rather than just a single taxon. The goal is to see how and which cascade effect happen, trying to untangle the complex interactions that take place in a multi-stressor context. While the main objective is to find the ecological relevant threshold for nitrates and MCPA in a multi-stressor context, I am also testing the effectiveness of a new methodology to detect sublethality effects on river biota. I am also hoping that my results will inform relevant stakeholders and legislators, guiding them towards implementing new scientifically based regulations by moving beyond the current framework, which consider pollutants in isolation rather through an holistic approach. Through this work, I'm hoping to balance both the need of agricultural community and the health of a functioning river.